This presentation will focus on genetic modification, and in particular looking at the genetic modification of E. coli to produce human insulin. Okay, so there's lots of different syllabus points about this particular area, and I'm just going to be focusing on the production of human insulin in this particular one. So firstly, gene transfer, why is it possible? So the universality of the genetic code is one of the pillars of biology. It says that actually the code is the same in all living organisms because we evolved from a common ancestor, that first ancestral cell. So this means if you transfer a gene from one species to another, the amino acids sequence that is translated will be the same whichever organism produces that particular protein. The way it's read is exactly the same in all organisms. This is because the DNA code is universal, and it means the same codons for the same amino acids in all living organisms. This is why genetic modification, transferring genes from one species to another, is possible. Now, the example that we've looked at so far is human insulin produced in bacteria. Hopefully, by now, you will have looked at the screen, the animations about restriction endonucleases or restriction enzymes, and also an overview of the process. But what this will do is just add a little bit more detail that you need to know. So firstly, instead of just like looking in the whole human genome and cutting out the gene for human insulin, this wasn't possible when this was discovered in the 1970s. So what they did instead was actually go to the pancreatic beta cells that produce the insulin and actually find the messenger RNA. Basically, if you look at a pancreatic beta cell, all of the messenger RNA within that cell, because it's so specialised, will be the messenger RNA for making human insulin. So you've got the code there already. And then what they used is an enzyme called reverse transcriptase. Now this is an enzyme taken from certain viruses, and it can actually convert RNA into DNA. And this is really good for a couple of reasons. Firstly, that RNA will just be the coding sequence for insulin. It won't contain any of the introns, it will just be the exons. So it will just be those coding sequences. And then this can be converted into DNA, which is going to go into a bacteria, because bacteria aren't very good at removing those introns anyway. They prefer it just in the exon state. So reverse transcriptase is used to convert that RNA into DNA. Now, the a plasmid, so that you can actually buy these plasmids, but you could extract them from bacteria. The plasmids will be cut with a restriction endonuclease, and the plasmid is what we call a vector. A plasmid allows us to move DNA from one species to another, so that's why we call it the vector. Then, complementary sticky ends are going to be added to the insulin gene. And the cut plasmid ends. Now, sometimes you can use restriction enzymes to create these sticky ends as shown in the animations, but in this case they can actually just add little sections onto the end of both the insulin gene and the cut plasmid ends, and they will be complementary, so it will slot in nicely and just fit nicely within that gap in the plasmid. Okay, then the human insulin genes needs to be actually secured in place within the plasmid, and they use an enzyme called DNA ligase. This is the same enzyme that is used to actually join up the Okazaki fragments in DNA replication. So that DNA ligase, it seals the phosphodiester bond and actually makes the DNA a whole piece. So that's what DNA ligase does. Then the plasmids can be inserted into the E. coli bacteria. Often they'll put more than one copy of the plasmid into each bacteria. So now we have our transgenic organism. And these bacteria can be grown in fermenters and stimulated to produce the human insulin protein. And then that human insulin protein will be extracted and purified. Okay, so this is an amazing technology. Before this process was discovered and developed, insulin was extracted from the pancreases of animals that had been, from the slaughterhouse. And the trouble with this was it was extremely expensive to produce, and also the insulin that was being injected into people wasn't pure human insulin. It was pig insulin or cow insulin. So using this human insulin produced by genetically modified bacteria really revolutionised the treatment of diabetes and allowed it to be produced in large quantities and at a much lower price. And also it allowed the production of that pure human insulin, which is much better tolerated by people who are treated for diabetes.